Okay, yeah. Hello, everyone. Thanks for sticking along for the morning session, early morning session on the third day of the meeting. Um, I wish I could have been there in person, but nonetheless, it's a great pleasure to be able to speak at this meeting again. So what I'm going to be talking about is episodic memory. So episodic memory is essentially the memory of like stories or events. So if I think of a particular story or if I think of something that happened to me, that's basically what episodic memory is. And I'll be talking about how a model for how episodic memory might be forming in the hippocampus. Um, Quick shout out to Collins. Thanks for the great talk yesterday, which covers a lot of the background of what I'll be speaking. So it makes my job slightly easier, but I'll try to keep this as self-contained as possible for people who weren't there yesterday. Now, if I'm going to be talking about memory to dynamical systems or physicist audience, I think one has to start, I guess, from here, from last year's Nobel Prize, which went to Hopfield and Hinton. And in particular, I'll be talking more about like Hopfield's work, and particularly what he did for which is known as Hopfield networks, right? So a Hopfield network is basically a model for content addressable memory or associative memory, where the essential idea is I want to set up some sort of system where I can recall a memory from some corrupted or partially observed state. So if I have some sort of saved state that looks like this, I want to be able to give you an input pattern, and the dynamics of the system itself should flow me to my saved memorized pattern, right? And Hopfield's beautiful work basically showed that you can formulate this entire problem as that of descent on energy landscape, where you have where different minima of my energy landscape essentially correspond to the memories that I want to be remembering. And so then if I initialize myself at some point, I'll then roll down this energy landscape until I arrive at a memory, right? So let's just look at this in a slightly more detail. So the way one sets this up is, so we have this kind of a state over which I want to be able to generate my various memories. We just think of this as some sort of network with all-to-all coupling. And we're going to give it some dynamics that look like this, where S of t plus 1 at the ith node is just summing in the inputs from all the other neighboring nodes. And then based on some threshold, it goes to some +1 or -1 state. Right? So it's very simple dynamics. You can think of this as being like classical Ising spins in like low temperature and so on. And the task over here is we want to now set up this dynamics such that the fixed points of the dynamics correspond to certain memories S mu. And what was shown basically is that if you set up your weight matrix, Wij, via Hebbian learning, basically as a sum of outer products of these memories that you want to remember, then this suffices. These S mu states become fixed points of these dynamics, and in fact, stable fixed points of these dynamics. Now, if you start doing this, though, you come across one issue, which is that there's a pretty sharp limit on the number of stored memories. So if I start storing various different S mu patterns, what one finds is that there is an upper limit of which is given by around 0.1 times the number of neurons in my system, beyond which I can no longer store any memories. And if I try to store additional memories into my system, I undergo catastrophic forgetting and I lose all the information. So one way to just describe that again in a slightly more graphical format, over here on the y-axis, I'm looking at the amount of information I can recover from this network. On the x-axis, I'm varying the number of patterns I store into the network. And so what one sees in a Hopfield network is that as you store more and more patterns, up to a certain point, you can recover order n bits of information, which is it's an n bit pattern, you're really recovering the entire pattern. But beyond that critical capacity, you go through a memory cliff where you forget everything. And this is obviously not at all like what biological memory is like, right? Like if I go and attend one more talk tomorrow, I don't want to, it can't be that I just forget everything that I've ever learned so far, right? So biological memory is definitely not like this. But it turns out that it's not just Hopfield networks, but there's been a variety of other different models that people have constructed over the years. They all seem to undergo different forms of memory cliffs, where in thermodynamic limits, once you store more patterns beyond a certain number, you just lose all the information. Now, interestingly, notice that all of these different memory cliff curves, they actually fit in an envelope. And this envelope is basically a theoretical bound on how much information you can expect to be able to store in a network. And this goes back to some older results that rely on some very classical statistical mechanics tools, which showed that the total information I can expect to be able to get out of a network like this basically scales as the number of synapses in my system. So if I have n nodes which are all coupled, it scales as n squared. And as I said, this is really not very biological. And so what we would like to have is, can we construct an associative memory model that actually spans this entire memory continuum, where as I try to remember more and more things, I never have a point where I catastrophically forget everything, but I might only gradually be forgetting things, right? And so in that context, one can say that the goal is going to be to describe a memory architecture that can flexibly vary the total number of stored memories without catastrophic forgetting. Now, I pitched this as being a useful goal when I'm thinking about biological memory. So perhaps one should take insight from biology and neuroscience and ask, what are the brain structures like that actually are encoding these memories? So it turns out that at least in vertebrates, the region of the brain that's known to be very important in the formation of episodic memories is the hippocampus. And so there have been some very classic experiments back in the 50s where hippocampal lesioning was done on some patients to prevent epileptic seizures, and it was found that these patients were unable to form any new memories. And so it seems to be that the hippocampus has an important role to play. Now, if you just look at the hippocampus in itself and just try to take one step back from just its memory aspects, and you ask, what does the hippocampus do? It turns out the hippocampus has two important roles. It has other things as well, but it has two important roles that I'll be focusing on for today, which is that it's relevant for the formation of spatial maps as well as episodic memories. So Collins had talked a little bit about this yesterday, but basically if you look at different cells in the hippocampus, you can see that these cells have spatial tuning, which is that they particularly fire maybe when an animal is exploring a particular region of the environment. And so in that sense, there's a very distinct spatial tuning in these cells. There's a capability of forming spatial maps in the hippocampus. And so in that sense, I mean, I first pitched a problem from the dynamics point of view about how do you build an associative memory that doesn't have this cliff issue. But this also leads us to a very natural neuroscience question, which is why should the same circuit of the brain be tasked with these two seemingly independent roles, one for spatial navigation and spatial maps, and the other for episodic memory? And so John O'Keefe has received the Nobel Prize for this back in 2014. And to really understand the spatial maps part of the hippocampus, we really have to also pay attention to the other winners of the Nobel Prize, who John O'Keefe shared the Nobel Prize with, which were Edward and Maybrit Moser. And so they had received the Nobel basically for their work on the discovery of grid cells. So the hippocampus basically receives very strong inputs from the entorhinal cortex, which is basically a neighboring region. And entorhinal cortex has a variety of very interesting spatially selective cells. And the one particular set of cells that fascinates me a lot is these cells called grid cells that were described yesterday as well. So here we basically have a top-down view of an animal exploring an environment. And whenever a particular neuron spikes, there's a white dot placed over here. And what we see is that this cell is basically sensitive to points that form a regular lattice, hence their name grid cells. And so this lattice basically has some length scales that range from around 30 centimeters to around 1 meter or so. And they're arranged in different modules in the entorhinal cortex. And in fact, I have some work that talks about exactly what how these different modules emerge. I won't have time to go into this. In fact, this is what I had presented, a rudimentary version of this work back in this meeting 2 years ago. I'm happy to say that it's come out just earlier this year. But I won't spend too much time on this. But what I will want to say just is the fact that grid cells overall can be modeled well through attractor models. And so I'll just describe a little bit about attractor models again, just a bit of a rehash from yesterday, because that'll be useful for my construction of an associative memory network. So let's just start off by considering like a sheet of neurons. So this is basically just a rectangular sheet. Think of this as a continuous set of neurons. And what I'm going to do is I have some neural dynamics where neurons are interacting with each other via some interaction WXX prime. And I'll assume that I have some sort of spatially invariant interaction between them. So for simplicity, let's assume it's a Mexican hat interaction. We showed in our paper that doesn't really need to be this restrictive at all. But let's assume it's a Mexican hat interaction. Then what you can kind of intuitively see is because of the excitation and inhibition, you have like some classic Turing pattern formation, and you have a nice hexagonal pattern that emerges on the sheet. Then if you can couple the motion, the flow of the pattern on the sheet with the motion of the animal, you basically get that if you probe a particular neuron, whatever patterning you have in the neural sheet gets replicated onto external space. And that's basically what gives rise to this hexagonal patterning in external space, right? And one other thing you can see from this is because of this two-dimensionally periodic structure of this activity pattern on the neural sheet, is if I now look at the very high dimensional population vectors of my entire sheet of neurons, all of these population vectors should live on some torus, right? And this has also been experimentally verified. There's been a variety of works that have really examined and experimentally verified this model for grid cells. Now, yesterday in the Great Talk, College described how he thought this was too simplistic and he wanted to make it slightly more complex. I'm going to go in quite the opposite direction. I'm going to try to simplify this one level further to then set up my associative memory network. One can always come back to this level of complexity from what I will describe later, but for a pedagogical purpose, it will be useful for me to simplify this a bit further right now. So what I'll do is instead of thinking of this entire periodic lattice over here, I'm just going to consider one single unit cell of my periodic activity pattern. So I'm just going to think of this kind of rhombus, which now has a lattice of neurons, and this activity bump over here is just going to be like one neuron that's like that's in some highly active state. And so then if I'm now just looking at this, instead of talking about the flow of neurons, I can just talk about moving this bump around on this rectangular lattice. And the other thing to keep in mind is that now one population vector, so one lattice like this, corresponds to one point on my torus. So if I move my bump around, I'll correspondingly be moving my point on the torus, right? And so this is just a simplified way of looking at it. I just have gone to like a discrete system to be able to think about this in a more analytic fashion. And I should really say that this torus over here is really a torus of fixed point attractors. So I can start off from some state that is not just a single neuron active, but I can have various neurons at varying levels of activity. And this will correspond to some point that's not on this torus manifold. But then if I just run the dynamics, it will basically pull me back to the torus manifold, and I'll go back to the state where I have a single active neuron. Right, so this is really my very simplified model of a grid cell module that I'll be considering for today. Right. So now, certainly there are multiple different grid modules that have been observed. So there are multiple different length scales over which the animal is tracking these periodic lattices. And so I'll basically think of that as something like this, where I have, say, maybe 3 different modules. Each of these is going to be a different lattice. And so total number of cells that I need to construct this lattice scale as the sum of lambda square, right? Where lambda is just the linear length of my periodicity. Now, because these modules are all acting independently, the total space that they can encode through a combination of these 3 modules basically scales the product of these grid periods instead of the sum of the grid periods. And so effectively what I really have is I have an exponentially large region of space that I can encode through this small number of grid cells, right? And so this is essentially going to form the crux of the spatial scaffold that I'll now be using to construct my episodic memory. But before that, I should also point out that because I now have this exponentially large number of fixed points that are actually laid out on a very well-structured manifold, which is just now unrolled out torus, which is just a very large 2-dimensional space, I can now construct very simple 2-dimensional velocity operators that I can inject into my system that will help me move from one particular fixed point to some other neighboring fixed point. So I not only have a very large number of fixed points, but I have a well-structured large number of fixed points. And then in the neuroscience community, this is basically what people think of as path integration, right? And so this is now set up the core ingredients that I need to set up my associative memory model. And so what I'm essentially going to do is I'm going to think of something that looks kind of like a tripartite architecture. So my first orange layer is simply my grid cells themselves. Then I'm going to simply do a random projection from grid cells to another layer that I'm going to for now label as the hippocampus, right? So it's simply a fixed random projection. Then the return weights, what I'm going to do is I'm simply going to learn them with Hebbian learning, just in the same way how Hopfield networks are trained with just 2 neurons that are coactive, you just increase the synaptic weight. So I'm just going to learn these backward weights through Hebbian learning. Now, crucially, none of the weights so far, the feed-forward projection weights or the backward Hebbian learn weights, none of these depend on the content of what memories I actually want to store. So if I ever want to encode any particular items, I'm not going to actually change any of this part of my network. This is a fixed scaffold. It's a fixed network. It just does its own dynamics. And then now whatever information I want to store into my network, I will present in this other layer, which is perhaps lateral entorhinal cortex or other parts of entorhinal cortex. And then I will simply perform Hebbian learning between this layer and my hippocampus layer, right? So I have one region that's a scaffold that is held fixed, and my other component that does heteroassociation of my information content with my scaffold. And that is basically why we call this model a hippocampal scaffold memory with heteroassociation. Now, before going further into what we can get out of this kind of a construction, I just want to draw a quick analogy. Is that this is almost like reservoir computing, right? I think some people in the audience might be familiar with reservoir computing, where the essential idea is of the philosophy of reservoir computing is that if I just have a fixed reservoir where I don't alter its weights, I can still get quite a lot out of its dynamics by simply training a set of readout weights. In that sense, what I've set up over here with grid cells and random projections to hippocampus is a scaffold, which is essentially a reservoir of fixed points. And now all I'm going to do is I'm going to train read-in and readouts from my reservoir of fixed points, and I'm going to argue that this is going to do very useful things for memory properties. So to really, and I should also really say that it's going to be this very same architecture that I'll be able to argue out in very small variations. They do not just memory of individual items, like what you would do in a Hopfield network, but also will do spatial memory and the formation of spatial maps. But it'll also do episodic memory and sequence memory, where you're trying to remember arbitrary stories or events. It's going to be the same core architecture that will achieve all of these different functionalities. So to understand this, let's just zoom in just to the scaffold part of the architecture first, and just see what interesting properties do we have over here, which will then be useful for setting up the rest of the memory. So what we have over here is basically just this grid cell and my hippocampus over here. And what we find is, because I had argued earlier that grid cells themselves have this exponentially large coding space, because you're getting a multiplication of grid periods, whereas you only have a sum of grid periods in terms of the number of cells. In that sense, what we find is that this collective network as well has a number of fixed points that grows exponentially with the number of grid modules. Now one could ask, okay, this is just in terms of the number of grid modules. Do you also need an exponentially large number of hippocampal cells? It turns out that we don't. We can actually prove exactly that the number of hippocampal cells needed to support this exponentially large number of cells only grows linearly with the number of grid modules. So in that sense, you truly have an exponentially large number of fixed points that we've set up with this random projection and Hebbian learning architecture. Now, if you're doing Hebbian learning on exponentially large number of states, one might ask, look, are we expecting the animal, before it can start using any memory of its own, to have to explore some very vast, exponentially large space before anything becomes useful? It turns out that's not the case. We actually, within this scaffold network, we have a very rapid generalization of fixed points, where I can actually explore an environment that scales linearly with the largest grid module. And that is sufficient to store memories corresponding to this exponentially large number of states. So really, in practice, a juvenile animal might only have to explore a region that's around 1 meter square to then be able to have set up its scaffold for storing a very, very vast number of memories. And if I have such a vast number of memories, one may ask, oh, are these like vanishingly small memories or vanishingly stable memories? That's not the case. Actually, all of these memories have very large basins of attraction, where I can put extremely large perturbations into my system and still recover the correct state. So one might be wondering, like, if I'm saying that I've set up a network field that does exponentially many fixed points, does this not violate the information bounds that I spent some time talking about, where I said that, oh, the amount of information you can store only scales linearly with the number of synapses or quadratically with the number of nodes? But the answer is no. Because what's happening over here is, so far, I don't have any arbitrariness in the fixed points that I'm generating. I can't store any particular patterns, any particular dog or cat. I have grid cells, which is just a predefined attractor. I'm just storing a predefined set of exponential fixed points. Or another way to think about a non-information theoretic sense, I mean, here I'm really saying there's no information to these memories. But the dynamical way to think about it is that the reason why Hopfield networks end up failing is that as you store more and more memories, you have a lot of spurious states that emerge, which are basically other minima of the energy landscape that don't correspond to actual memories that you want to store. Now, in this system, because of the extremely symmetric structure of the grid cell coding space, it turns out that every spurious fixed point that forms corresponds to some other fixed point that you actually wanted to store. So in that sense, you're actually using all of these spurious states, and so you don't actually have any dynamical issue with storing more and more patterns in the system. So in this sense, we basically arguing that this entorhinal hippocampal network generates a robust scaffold with an exponentially large number of fixed points. Now what I can do is I can now take whatever information I want to remember and just tag it onto various fixed points that I've already set up. So if I have any particular memorized pattern, I'll now inject this into the hippocampal layer, it'll get cleaned up through the grid attractor dynamics, and then I'll get some recovered pattern that is the cleaned up version of what I started off with. And one can now explore what happens in this network as I store more and more patterns. And one indeed finds that for a small number of patterns, I have perfect recovery. As I increase the number of patterns, I have a gradual decay of information. And when I have a lot more patterns as well, I've still not quite lost everything about my initial pattern. So I only have a very gradual decay of information. And moreover, we can actually prove that this gradual decay is asymptotically proportional to this theoretical upper bound of how much information one can expect to store in a network. So we only have these smoothly degrading memories. And another way to really put this information is I can say, let me look at how much information I can get out of the system, normalized for the number of synapses. And so what we see in our model of vector hash is that this asymptotically approaches a constant that we can prove. Whereas for all other memory architectures, whether it's Hopfield-like networks or all other variants of Hopfield networks, they all undergo this memory cliff where you've just lost all the information once you stored beyond a certain number of patterns. Now, an interesting thing that might appear to you is that, if you look at this network and you just unfurl it, it kind of looks like an autoencoder, right? So one could ask, okay, if I set up an autoencoder as an associative memory, how would that do? It turns out that that's also quite bad. That's this curve over here. And so in this sense, what we actually found over here is a setup where backpropagation or gradient descent is unable to find a good solution. But instead, if I hard-code representations from neuroscientific insight, I actually get much better performance at this task of storing memories. And so really, we've got this scaffold that stores arbitrary inputs. And the qualitative picture for you to keep in mind, really, the distinction between how a Hopfield network fails at this and why our network succeeds at this, is that in a Hopfield network or other related associative memory architectures, you have the same set of neurons that is tasked with both the content of the pattern that you want to remember, as well as stabilization of the patterns through its recurrent dynamics. Whereas what we've done here instead is we factorize this task of generating stable fixed points and stabilization of fixed points from the actual content of the information that we want to remember. So it's almost like we've constructed like a clothesline, and now whatever information you want to remember, you can just pin it onto this clothesline at various points, and you can just keep reusing this clothesline for whatever arbitrary memories you want to store. So this is how we're essentially able to store independent items. But let's talk more about spatial memory and episodic memory as well, right? So as I said earlier, that now because these are very low, these fixed points live on a low dimensional manifold, I now have a simple velocity input that I give into my system that then makes me transition and helps me pick which fixed point do I want to store my next memory onto. So I could, in principle, just have been arbitrarily choosing points on my clothesline, but now with a velocity input, I can systematically choose which point on my clothesline do I want to hang my next piece of cloth. And so what that means is that now if I have some environment where I'm observing some landmarks, I can now zero-shot try a new trajectory, and I'll be able to tell you where those landmarks are, because I've indeed been able to construct a very faithful representation of landmarks to spatial locations. And this is just like one or two rooms, but because I have this very large coding space, I can store several rooms in my very large coding space. And I won't spend too much time talking about all the other experimental results that you can compare with over here, but really you can look at the kind of hippocampal representation that you might expect, and we find a very good agreement with what we get out of this model. So this tells me how I can build these spatial maps and spatial memory without catastrophic forgetting. But now let's next get to episodic memory, right? So episodic memory, what I told you earlier was episodic memory is essentially the memory of sequences or events, right? So for me to be able to remember episodic memory, which is an arbitrary sequence of some sort of sensory observations, I first need to think about how do I get sequences in this model, right? And I'm going to take the same philosophy of factorization, the factorization of the task of content from, stabilization to try to approach this problem as well. And so what we do over here is we have some very long trajectory in my extremely large space. And what we basically say is I'm just going to learn a mapping from my hippocampus state vectors to just this two-dimensional velocity vector of where I need to go next. So I'm tasked with a very low information problem. I only need to learn a low dimensional velocity output. And we find that this simple architecture can actually remember this very, very long sequence. In contrast to the same sequence, if I try to learn it via some sort of associative memory techniques, Hopfield-like or other, Hopfield-like models, we find that we just learn vanishingly small sequence. And in fact, in this context, like sequence models actually have even worse capacity than the 0.14n for the regular capacity of Hopfield networks. So in this sense, because we realize that this very low information task that we can reduce the problem to, we can now store arbitrary sequences. And so now we just push for the same idea. And now I can tag my arbitrary sequences with different sensory observations that I want to remember. So if I now have some particular sensory observation that maybe corresponds to some certain things that I'm remembering, I can just tag these onto different points of the sequences, and I can use this setup to now remember these items. I should crucially point out that while I set up grid cells by thinking about spatial maps, there's nothing that's really constraining me to be only thinking about spatial maps anymore. I can take the same grid cell architecture, I can be giving it velocity inputs corresponding to some completely abstract context as well, and I can still generate sequences, and I can still store information onto these sequences, right? And we can again do the theory here as well, and we can find that the amount of information that you can recover from the system decays with the length of the sequence. I don't have any catastrophic forgetting, and once again, asymptotically proportional to the bounds that one would expect from a theoretical, very abstract formalism. And just again to contrast this with recurrent associative memory architecture, what you basically have over here is that you very rapidly fall into different spurious states, and where you should be recovering some birds or some like a toucan or like a snake or something, so you're just recovering just garbage spurious states instead. And we can again quantify this in terms of the mutual information per synapse and so forth, and we find that once again we asymptotically approach a constant, whereas all other architecture models seem to just fall flat at a memory cliff. So the qualitative picture here really is that if I think of the general task of episodic memory, which is where I want to remember a sequence of events, as such, my sensory observations live in very high dimensional space. And remembering transitions in high dimensional space becomes a very difficult task. It's a high dimensional learning problem. And there are lots of possible directions you need to go to. But instead, we can utilize the spatial scaffolds, the fact that grid cells live on these nice low dimensional, these two-dimensional tori, and we can utilize that fact to then reduce my problem to just learning two-dimensional transitions or very low-dimensional transitions. And that really significantly simplifies the task of episodic memory learning. And this is really how we're able to utilize the spatial scaffolds of the hippocampus and the internal cortex to be able to generate good episodic memory. Now, okay, so hopefully this kind of answers the two big goals that I had set up with initially, right? How do I build an associative memory architecture that doesn't have a memory cliff? And why should the same hippocampal complex be tasked with both spatial navigation and episodic memory? But it's a very reasonable question to ask is, what I've set up over here, is this meaningful in the context of what's actually going on in the neuroscience? Or is it just inspired by the neuroscience in terms of, oh, I'm going to put in grid cells over here and some hippocampus and see what happens, right? And so one can ask, does this actually recreate other phenomena that we didn't put into the system to start off with, right, in a sense? And so what we basically get is that this model can recreate a variety of hippocampal phenomenology, which ranges from all sorts of different cell-cell correlation results, these relate to lots of older grid cell results as well. But you can see that, cell-cell correlation between grid cells is preserved, between hippocampal cells is not preserved. I won't spend too much time boring with all the details of all these comparisons, but when we talk about different qualitative comparisons about, how quickly should maps emerge when you, say, turn on the lights in an environment, or what happens if you cut off certain regions, or if you cut off certain synapses and you ask what happens to the maps. Or what kind of correlation structure do you expect putting grid cells and place cells in certain rooms and so on. And you can do all sorts of different things, and all of these qualitatively match what you get in experiments. Not just this, but you can also talk about other more general phenomena. So in hippocampus, people often characterize different kinds of cells, splitter cells, which depending on which lap of a maze you're in, you might have cells that distinguish representations, or you might have task-dependent cells or directional cells and so on. So you can have a variety of different cells that people have found in the hippocampus, ascribing them to various different things. But we find that in our model hippocampus, which is simply coming from this very simple idea of random projections from grid cells, with this feedback recurrent loop, can actually recreate all of these different kinds of observations. We can actually do the different kinds of tests that people have been doing over the last several years in these experiments, and we can find that vector hash actually does recreate this kind of phenomenology. So really, in that sense, we are able to get out a lot of interesting properties in the system. Now, we're doing all of this. We realize that another thing that is very directly related to spatial navigation and memory is this whole idea of memory palaces, right? So what are memory palaces? So memory palaces are basically this idea where you have all these memory competitions where I'll just shuffle a deck of cards and I'll give it to you and I'll ask you to memorize the exact sequence of the deck of cards, which is certainly a very very difficult task. Now, what's been found is that what a lot of the memory athletes do, a lot of the very successful ones, is that they will imagine something like maybe their childhood home, and they will place the cards at different locations on in their childhood home. And then instead of just trying to independently remember these cards by themselves, they will imagine walking through their childhood home and picking up their cards as they go. And that turns out to be a very successful strategy for remembering arbitrary things. And this seems kind of paradoxical, right? Because it seems like you've burdened your memory task further by not only remembering the sequence of cards, but also now remembering it in the context of some other spatial environment. But how does burdening this task further actually help you out, right? And so we actually found that you can actually explain this very well with our model, where you can now use the sensory states themselves, the sensory reconstructed states themselves, as their own memory scaffold to then now store arbitrary, completely independent items. And so what you actually get is if you now form an association of this nature, is that the memory palace items, the items that you had to remember as a part of this memory palace challenge, you can actually remember them with extremely high fidelity, even if you are unable to remember the sensory states with complete fidelity. So if these are now my sensory landmarks in my environment, even though they might decay with increasing lengths of sequences that I'm trying to remember, the particular items that I want to remember as a part of my memory palace task, those actually are unaffected and they have much higher fidelity over which you can remember them in sequence. So I'd just like to conclude with that. It's just to say that I presented an architecture that is a hippocampal entrained model that recreates both item memory, spatial mapping, and episodic memory. It crucially uses a representational structure used in spatial mapping to generate this high capacity episodic memory. And in doing so, it then avoids catastrophic forgetting and these memory cliffs that other associative memory architectures are quite distraught with. Um, in setting up this model, we happen to independently recreate multiple aspects of seemingly disparate hippocampal phenomenology, and we can also explain these aspects of the method of loci and memory palaces and so on, using the familiar paths as their own memory scaffold onto which you can tag more information. So with that, I'd just like to quickly acknowledge collaborators and mentors. Of course, Ela at MIT, who I've been working with for the last several years. Sue and Mikhail are grad students in the group. Rishi was an older postdoc from Ela's lab, who's now at UC Davis. Most of what I've described today is really in the first paper over here. But really, these two papers over here, both of which came out a few months ago, really summarize all of the results that are described in my talk. Before ending and going to questions, I just want to also have a quick shout out. I will be starting a group at ICTS soon. If there are people who are interested, do reach out to me and get in touch. Happy to talk further. Not necessarily just about problems about hippocampus and spatial navigation, but even generally computational neuroscience. I'll be happy to talk to people. With that, I'd like to thank everyone and take any questions.
Yeah, so we have time for just one question. But before that, I have a question for you.
Yeah.
Okay, we'll have a session for open problems at 11:10.
Mm-hmm.
Would you like?
I will join that. Yes. I'm enough of a midnight owl to join that.
Hi, Sarthak. Very nice talk. Lovely work. So, I just wanted to talk about your connection between reservoirs and the entorhinal network, entorhinal CA1 network. So, can you can you tell us a little bit more about, so in reservoirs, I guess the reservoir layer doesn't have to have the same kind of topographic organization that you need when the fixed points are topographically organized
Yes.
in this. Does this act as a constraint, or does it make things easier for you in terms of memory capacity? So if you had an arbitrary reservoir, an arbitrarily complex reservoir, would you be in a position to store a lot more? And does topographic organization place constraints on that?
So that's a great question. So in fact, this is something we've actually explored. So if instead of considering this, very well structured grid cell torus attractors, let's just think, as a thing. Let's say I set up a reservoir with just random fixed points in some very complex fashion. I don't put any topography in it. There are some things you can continue to do, and there are some things that you lose out on. So it turns out that the memory capacity that you get in terms of this item memory, particularly this asymptotic bounds, this doesn't strictly rely on grid cells as being this particular topological object. So if you have an arbitrary reservoir, you will still get good item memory. But the thing you lose out is that you no longer have the capability of now giving a low dimensional velocity update into my system to help me transform between one fixed point to another. So the high level answer essentially is that you are great at item memory. You don't lose anything. Asymptotically, you don't gain anything. In terms of proportionality constants, it's a different matter. You can get better proportionality constants, but asymptotically, you don't gain anything. But you lose out on a lot of the sequential results because you now no longer have low dimensional operators that act on these fixed points.
Yeah, I think it's my question is sort of related. Okay. Yeah, maybe I can just. So what's the when you initialize from input from hippocampus, from whatever limited understanding I got, so the grid cells would sort of collapse into one point, right? One of them, one fixed point, one unit getting active eventually. So what does the dynamics offer here? I mean, why do you need a dynamics? You can always map it to a single point. So how does the dynamics in the network help?
So the dynamics are really what are doing a lot of the error correction for you. So the idea is that I'm using the grid cell attractor to actually do all of my error correction. So when I have some sort of noisy state, it gets mapped to some noisy hippocampus state, which goes to some noisy off manifold point in my grid space. Then I'm using my grid cell manifold attractor dynamics to actually pull me on manifold again, which helps me recover the cleaned up state. So the dynamics are really what's doing the error correction.
So, but I mean, you cannot create boundaries partitioning those errors in the hippocampal input itself as a sort of classifier. I mean, the grid cell can do a better classification of that error space to same point mapping. I mean, I don't understand how.
Yes. Yes, in fact, I am very specifically arguing that it can do better. So one could consider an alternative, and we have this in the appendix of the paper, where basically you say that, maybe at the hippocampal layer itself, maybe I threw some recurrence or through some feed-forward architecture. I want to do some kind of error correction, right? And what we actually argue out is that this particular architecture where you have this random projection and your return Hebbian weights actually gives you significantly stronger error correcting properties. And in fact, if you set it up in the way you're describing, where you use the hippocampal layer in itself, to try to give you the error correction, one can actually show that you will get a memory cliff again, and you'll have a very sharp cutoff on the amount of information you can store.
Yeah. So we'll say bye to you now, Sarthak. And unless you'd like to stay for our fun stuff, but I don't know how we can technologically manage. So our next speaker is Arpan Banerjee.
I can stop sharing.